Next up, we have Dr. Giada De Palma, who is an assistant professor at McMaster University, specializing in microbiome research and its interactions with the neuroimmune system. Giada completed her PhD at the Institute of Agrochemistry and Food Technology in Valencia, Spain, under the supervision of Dr. Yolanda Sanz, where she investigated the role of gut microbiome in celiac disease. She then moved to Canada for a postdoctoral fellowship on the microbiota gut-brain axis with Dr. Burshik and Collins, with whom she continues to work to this day. Her work focuses on translational research, exploring the gut microbiome's role in disorders of the gut-brain axis and mental health conditions. She has a particular interest in microbiome-diet interactions, microbially-derived metabolites, and their impact on host physiology and symptom presentation. Recently, Giada and colleagues were awarded the Weston Foundation Proof of Principle Award for translating research into practical applications. And today she'll be presenting her talk entitled The Role of Gut Microbiota in Pain Transmission, and very much looking forward to it. Take it away, Giada.
Thank you very much for inviting me to speak today. Let me share the presentation. Thank you very much again. It is a real pleasure to be here today. And I don't think I really have to tell this audience what the microbiome is and how important it is for host health and homeostasis, especially because Brittany gave such a nice introduction on the gut microbiome. And we know that microbiome is central to many physiological functions, and some of them are depicted here. And we know this because, and thanks to studies on germ-free animals or notobiotic animals. These animals have been shown to have physiological metabolic abnormalities compared to conventional animals, to have an imbalanced immune system, to have an altered enteric nervous system, an altered behavior, brain biochemistry, and we heard about this, altered gastrointestinal motility, and altered pain perception, among others. And we also wanted to look, as other groups have done, into the role of gut microbiota and visceral pain. And to do that, we also use germ-free animals and SPF conventional animals and tested their visceral pain. We did that using the colorectal distension, which is a test where we insert a balloon up the colon 2 centimeters, and then we inflate the balloon to 3 levels of distension. You can see here 100, 200, and 300. And then we measure the abdominal contraction in response to these distensions. And you can see here representative traces of those responses. And so what we observe is that germ-free animals, especially females, present with increased visceral motor responses to colorectal distension, and you can see here. And as well, when we looked, and the study was led by Dr. Julien Pougeot in our group, when we looked at a neural peptide that is involved in pain neurotransmission, CGRP, we found that CGRP production by DRG neurons was greater in germ-free animals, especially in females. So when we think about pain, we normally think about visceral hypersensitivity. And for visceral hypersensitivity, we refer to that exaggerated pain signaling in response to stimuli within the visceral organs. And this can be either visceral hyperalgesia, which is an enhanced sensation to painful stimuli, or visceral allodynia, which is a painful sensation to innocuous stimuli. And visceral hypersensitivity is the hallmark of a disease that we study a lot in the lab, which is irritable bowel syndrome. This is a chronic disorder of gut-brain interaction characterized by recurrent abdominal pain, visceral hypersensitivity, and altered bowel habits. And the underlying mechanisms to visceral hypersensitivity in IBS and in other disorders that present with visceral hypersensitivity are multiple. These can be central, and I want to touch upon these, and there can be peripheral mechanisms. Among the peripheral mechanisms, we can have, for example, an increase in excitatory pathway during ongoing inflammation, or we can have, after the resolution of inflammation, the sensory afferents that are still hypersensitized, and this is the case, for example, of post-infectious IBS. Or we can have an increase in excitatory mediators, such as serotonin, histamine, prostaglandins, and proteases, among others. So, talking about host mediators, and Giovanni Barbara at the University of Bologna already 2 decades ago showed that in the colon of IBS patients, we can find an increased number of mast cells. You can see here, it's clear in this picture. And as well, these mast cells are in close proximity to nerve endings. And these arrows point to the nerve endings. When they quantified the mast cell mediators such as tryptase, here, histamine and prostaglandin, they found that they were increased in IBS patients in comparison to healthy control. And so there, the conclusion was that perhaps colonic mast cell infiltration and their colocalization with the neural fibers contribute to visceral hypersensitivity, abdominal pain in IBS. And so we wondered whether the microbiome, sorry, I need to move the bar here from the Zoom. There we go. We wonder whether the microbiome may play a role in modulating this mast cell accumulation in the colon of the patients. And this is because the microbiome has been implicated in the expression of IBS as patients present with altered gut microbial profile and microbial metabolic activity. And so to study the role of microbiome and mast cells in IBS, we use a translational approach and took germ-free animals and colonized them with the 2 samples of IBS patients or healthy controls, and then looked at their mast cells. And in that study, we confirmed what Giovanni Barbara had shown almost 2 decades before, that IBS colonized mice present with an increase in mast cells in the colon, and these mast cells are close to the nerves. And as well, we observed that the microbiota in this IBS colonized mice, here in red, was in close proximity to the epithelium. And in fact, when we looked better, we found that the microbiota in blue now came in close to the mast cells in pink here, and this is the quantification. Through a pharmacological in vitro experiment, we also show that the IBS microbiota drives mast cell migration through the activity of the histamine receptor 4 and the granulation through the activity of the toll-like receptor 4. Thus, we concluded that this can contribute to the gut dysfunction and the visceral hypersensitivity that we observe in the IBS patients. However, so we proved the point that the microbiota is able to drive mast cell accumulation in the colon of these mice, but however, it was not clear how the microbiota was doing this. And so we started thinking, and we turned our attention to a study that we did few years ago already, where we showed that transplanting the fecal microbiota from patients with IBS and diarrhea predominant was not only inducing faster intestinal transit in the mice and impaired body function and the low-grade inflammation, but was also altering the serum metabolome in these mice. And this was got us thinking, because despite the unremarkable difference in the microbiome composition between these IBS colonized mice in red and these healthy colonized mice in blue, we saw a striking difference in the serum metabolome of the mice. You can see this is an unsupervised analysis of the metabolome, and you can see a complete separation between the 2 groups. And as well, when we did the supervised analysis, we saw a nice separation in 3 subgroups in the IBS colonized mice. So this meant that the microbial composition was not so important, but it was the function. And of course, we just heard about how important it is, what the microbiome produces. And how can we manipulate microbial function in IBS? So a very good way to manipulate microbial function is through diet. And diet is a major manipulator of microbial composition and function, and is also the key, and microbiome is the key mediator of the physiological effect of diet. And we know that diet in IBS is important because IBS patients often report food-induced symptoms and make dietary modification to manage those symptoms. And we also know that IBS patients often adopt a gluten-free or a low fermentable carbohydrate diet, but the underlying mechanism to their efficacy are not clear. And just opening and closing a parenthesis, of course, there are central mechanisms to this diet microbiota interactions in IBS and their efficacy. And as it has been shown that increased symptoms to fructans in IBS are associated with altered brain responses in pain-related regions. But beside these central mechanisms, we wanted to focus on the peripheral mechanisms. And of course, we thought about a study that we did a few years ago where we put IBS patients on a low fermentable diet and found that with this low fermentable diet, we had changes in the gut microbiota profiles and urine histamine levels. You can see here after the low FODMAP, urine histamine goes significantly down in a subset of patients, and as well, abdominal pain scores significantly correlated with histamine. And histamine, of course, caught our attention because is a key neuroimmune mediator not only produced by mast cells, and we heard about the importance of mast cells in IBS, but it can also be produced by bacteria. And so we hypothesized that the gut microbiota contributes to visceral hypersensitivity through the production of histamine. Again, to look into this, we took another translational approach and colonized germ-free animals again with the 2 samples of patients with high urinary histamine in red, IBS-HH, low urinary histamine in blue, IBS-LH, or healthy controls. We did a battery of in vivo and in vitro assessments and kept the mice during the duration of the study, either on a high or a low fermentable carbohydrate diet. And this was to modulate with diet the histamine production by the bacteria. The first thing we looked at was, of course, abdominal pain. And in this case, again, we looked at the visceral motor responses to colorectal distension. And we observed that the IBS-HH colonized mice in red presented with an increase in visceral to both volumes of distension, and here you can see the representative traces. And as well, we found that the mechanosensitivity of colonic afferent nerves was increased in these IBS-HH colonized mice. Then we looked at the mast cells again in these mice and observed that the mast cells were increased in the IBS-HH colonized mice. And again, the mast cells were in close contact to nerves. You can see here the quantification, and here in green are the PGP 9.5 nerves. And this is again reminiscent of our study and of Giovanni Barbara's seminal studies. So again, we show that the bacterial histamine drives mast cell accumulation in the colon, but we still didn't know what was mediating this accumulation. And so we looked into histamine receptors. And again, histamine receptor 4 was what was mediating the accumulation of mast cells in the IBS-HH colonized mice, as you can see here. And histamine receptor 4 has been shown previously to have a higher affinity to histamine more than the other receptor, and as well as been implicated in visceral hypersensitivity in other models and in pain. And when we when we use an antagonist against H4 receptor, we see a decrease in mast cells in the colon of these mice, and as well as significant decrease in visceral motor responses to both volumes of distension during the colorectal distension. So now we just wanted to know what was in the microbiota driving this mastocytosis in these IBS HH colonized mice. And so we tested the microbiota for the production of histamine from histidine, which is the precursor of histamine, and found that the IBS HH colonized mice microbiota and patients was able to produce high amounts of histamine in comparison to the other microbiotas that we tested. And as well, we went on and identified the major producer in these patients and mice, and found that Klebsiella aerogenes, a gram-negative bacterium that possesses the enzyme histidine decarboxylase, which is able to convert histidine into histamine, was the major producer. So we genetically manipulated Klebsiella to lose the capacity to produce histamine and monocolonize germ-free animals. And you can see here that when we colonize the mice with this Klebsiella aerogenes that no longer is able to produce histamine, we see a significant decrease in mast cell accumulation in the colon in comparison to when we colonize mice with the original isolate or other unrelated Klebsiella species. So now we prove that bacterial histamine was driving the mastocytosis in these mice. And then we went back to our original patients from that original diet study that we did a long time ago and looked at Klebsiella aerogenes abundance in those patients and found that Klebsiella was actually higher in the high urinary histamine patients, and it was lower when the patients were on a low fermentable diet. And when we looked into a different data set from a published paper from the group of Dr. Kashyap, we found that Klebsiella aerogenes was actually higher in the IBS patients from this cohort, and the HDC, the histidine decarboxylase gene, was also higher in these IBS patients, confirming the results we had seen in our study. In the original studies, the patients were put on a low fermentable diet and the urinary histamine was decreasing. So we put the mice on a low fermentable diet and saw a significant decrease in histamine production from this bacterium. And as well, we saw a decrease in at least one of the two volumes of distension during the visceral motor response, during the colorectal distension. When we looked into the possible mediator of this effect of the low FODMAP, we turned our attention to lactic acid, which was significantly decreased in the IBS HH colonized mice. And as well, lactic acid bacteria was significantly decreased in the IBS HH colonized mice on a high fermentable diet. And this was coming back up when we put the mice on a on a low fermentable diet. And this made sense because the histidine decarboxylase from gram-negative bacteria are highly susceptible to low pH. So a lactic acid would be enough to lower quite significantly the luminal pH and so deactivate the HDC and limit the growth of Klebsiella aerogenes as well. So in this study, we concluded that microbial histamine induces visceral hyperalgesia through induces visceral hyperalgesia through inducing an accumulation of mast cells in the colon through the activity of the H4 receptor. This accumulation of mast cells in the colon results in an increased secretion of histamine, and as well, this histamine goes and sensitizes sensory neurons through the activity of the H1 receptor. We showed that that fermentable carbohydrates modulate the production of histamine by this microbiota, and as well, lactic acid bacteria are able to decrease the capacity of the bacteria to produce histamine. So many questions remained and remain at the time now. And one of the questions we try to answer is whether mast cells are necessary to observe the effect that we observed in these models. And so whether perhaps histamine from the bacteria could directly sensitize the neurons and bypass the mast cells. And we found that indeed mast cells are necessary. And this is a mast cell dependent mechanism because when we stabilize the mast cells in these IBS HH colonized mice, we do not observe, we see a decrease in visceral motor responses, so a decrease in visceral hypersensitivity in these mice. And sorry, I should add that we are also doing experiments in mast cell knockout mice to confirm this result. So, but beside these preclinical studies, our interest is really to move this data from the bench to the bedside and so to clinical practice. And so to do that, we design a clinical trial that now has been closed, and we are analyzing the data in collaboration with our longtime collaborators at Queen's University, where we recruited 30 IBS patients with recurrent abdominal pain, moderate abdominal pain, and we randomized them to 3 challenges, each separated by 1 week of washout. These are the 3 challenges. We had a high FODMAP beverage plus a placebo plus L-histidine. L-histidine is the precursor of histamine. We had a low FODMAP beverage plus a placebo plus L-histidine again, and a high FODMAP beverage plus a probiotic plus L-histidine. And the idea here was to try to modulate symptoms and histamine production in these patients and try to understand a little bit better the underlying modulator of histamine production in patients. And the outcome was the levels of histamine and the metabolites present in urine and stool samples from IBS patients. And the secondary outcome was microbial histamine production, symptoms worsening on the high FODMAP diet, and changes in dietary intake. So while we are still analyzing the data, what has become very clear is that, however, I would really like it to be just histamine, it would seem that things are more complicated, and perhaps we should focus as well on other histamine-related metabolites, such as N-acetylhistamine, which has been reported to be produced by gut bacteria, and as well those products from the, excuse me, the diamine oxidase activity that produce these other metabolites from histamine. And one of them, imidazole 4-acetaldehyde, has been recently shown to be altered in IBS patients as well. And for that, please stay tuned, and I hope I can show you soon the results of this clinical trial. And in my last few slides, I just wanted to show some data that we looked into thanks to the pan-Canadian study Imagine, where IBS patients, healthy control, and also IBD patients are followed longitudinally during 4 years, and fecal samples, symptoms, and various questionnaires are collected. And so we were lucky that we could access some of these samples, and there we wanted to look into the histidine decarboxylase gene variants and the diamine oxidase gene variants. So histidine decarboxylase produces histamine, and diamine oxidase takes histamine and produces these catabolites of histamine, and as well here. And as well, we wanted to access the pain scores. So we had access to some of these patients and healthy controls, and we looked at these genes and the pain scores, and we did that by using a Poisson model with a negative binomial model with an offset for sequencing depth. And there we found that the IBS patients presented with an increased gene variant, HDC gene variants, in comparison to the healthy control, but they presented as well with a significant increase in DAO gene variants in comparison to the healthy control. And this was not exactly what I had hypothesized, but we are looking into it better. And as well, we found that when we split the patient into high IBS with high pain and low pain, we found a significant difference for both HDC and DAO gene variants. And again, we found significantly fluctuation in the gene variants of HTC and DAO depending on the IBS subtypes. So all this means that things are so much more complicated than that we originally thought. And we have to look into this better to understand in which subgroups these gene variants, and especially the bacteria that harbor them, are playing a role with abdominal pain. And in fact, when we looked just at the broader taxonomy for the HTC gene, we could see that many bacteria possess this enzyme, but mostly proteobacteria, as we observed, and some firmicutes. But this is the exact same for the DAO genes. So now we have really to look into which bacteria in the IBS patients and the healthy control harbor this gene, and whether there are differences between healthy control and patients. So this leads me to my take-home message, which is that GI visceral sensitivity can be shaped by an individual gut microbial community. Aberrant microbial metabolism might underlie symptom generation in a subset of patients with IBS, and that the manipulation of microbial function through diet or microbial-directed therapies appears to be a viable option in a subset of patients with IBS. And with that, I would like to thank all the people that contributed to this work, our collaborators at Queen's University, of course, our funding agency, and you for your attention. And I'm happy to take any questions.
All right. Excellent, Giada. That's quite a tour de force as well. I mean, so many translational studies from the bench to the bedside. Really, really cool to see. We have no shortage of questions, so we'll try to get through a couple in the time we have remaining. First up, lower FODMAP also results in less bloating and less bloating-induced pain. Is the histamine effect independent of gas production? Might the bloating promote an immune response independently that might promote sustained sensitivity?
That's a good question. I mean, often they go together, right? These low FODMAP are prescribed to decrease bloating and gas, and often we see that there is a correlation between abdominal pain and gas, so it is possible. But in our case, we only found correlation with the abdominal pain mostly, and we think it could be coupled with the bloating because the two things go together. We think it's mostly for abdominal pain.
Kind of as a tack-on question on that, do you think fermented foods might have any positive effect here? Maybe it's removing the fermentation from happening in the gut. So if you were to consume them outside, you might have some positive effects.
Possibly some fermented food actually include lactobacilli species, right? And we saw that lactobacilli are actually able to lower the pH and have an effect on Klebsiella, and Klebsiella, in this case, Klebsiella just as a microbe model to use as a histamine producer. So it is possible. We also know that lactobacilli produce low level of histamine and can also activate histamine 2 receptor, having an anti-inflammatory effect. So it's possible.
Okay, cool. I think we have time for one more quick one. How about this one? Do the IBS patients often have a history of recurrent type 1 hypersensitivity reactions, such as allergies?
Often these patients have lots of comorbidities, and so sometimes they have asthma or they have some allergies, but it didn't come out when we looked at the subset of patients that had this histamine producing microbiota. It didn't come up like this is the profile of patient that we should look at. So it happens. It is a comorbidity, but it's not clear that this is a co-occurrence all the time.
Gotcha. Oh, very interesting. Definitely a good subject for more research as well, right?
Yeah.
Well, excellent. I guess with that, I would like to thank both of our speakers again, Giada De Palma and Brittany Needham, for some excellent talks. Right. Thank you, everyone. Bye.